Thank you very much. Thank you, Rachel, and thank you for coming. So today, I'll talk to you about low-cost microfluidic devices and how to detect environmental pathogens. And I have a new subtitle, which is, does this have anything to do with the carbon cycle? And I hope I can convince you at the end of this. If you're not convinced, it's okay. We can fight it out in the end. So first, I'll take you through how I got to be here today. So Rachel kindly asked to review my CV. So first, I did an undergraduate in biochemistry and chemistry, where I got to do both molecular biology, but also some core physical chemistry. So I did some electron spin resonance and studied liquid crystals, and I'm really happy that my supervisor at the time, Professor Jeffrey Lockhurst, is here in the audience today. Then I did a PhD in biophysical chemistry, so I got to study how lipids and how they interact on the membrane of cells. And this one?
Press the top thing. Or
Top thing?
Yeah.
Perfect. So how they affect enzymes that reside both in the cytoplasm but also on the membrane of cells. Then I found that a bit theoretical, so I wanted to move to more applied things. So I was really lucky to be able to move to a postdoc at the National Oceanography Center at the NERC part, so the government-run part. And I was really lucky because I was involved in the Lake Ellsworth project. This was in the sensors group, so the PI there is Matt Molan. So we got to develop technologies that were deployed in a subglacial lake in Antarctica. This is ongoing, so they're going again. You might have heard about this on the press. So basically they didn't actually drill through the ice. They were meant to drill through 3 kilometers of ice and then get samples out of a subglacial lake. So they're going again. It's going to be amazing. And we used the same technologies that NASA used to decontaminate aircraft before they go off to space. And also, I got to do viability assessment. So if there's actually anything left on the instruments before they're deployed in such a pristine environment. Most importantly, when I was there, I started to work with microfluidics, which is a thing I really like, and I'm going to talk to you about today. And that I'm doing at the Institute for Life Sciences, which was founded in 2011 at the university. It was a really big investment. It's a really nice place to work. It's really new. The labs are really well equipped. And the best thing is that it houses scientists from across the disciplines. And although I started at biological sciences, I'm now part of electronics and computer science. So basically, this is my guide, how to do from biology to electronics and computer science in a few years. At this point, I ran out of schools, so basically they wouldn't have me in archaeology, so I had to write the fellowship and go somewhere else afterwards. More on this later. The fellowship, not the archaeology. There's no more of this. So I'll try and tell you why do we need to measure organisms in water. And here's where the carbon cycle comes in. So the global carbon cycle is a very complicated system. And it's a way scientists have explained how carbon flows within the globe, basically. There are many components, so there are terrestrial, atmospheric, and oceanic components. But a very big part of all of this is the biological pump, which consists of all the living components of this. So basically cells. And there are different types, especially in the ocean component. So first, there's phytoplankton that are very important molecules because they produce most of Earth's oxygen. There is a debate, so they make a lot of oxygen. It's 50 to 90, so there's like a massive error bar there, but they make oxygen. And they lie at the base of the marine food web, and they're primary producers. So they start making a bit of energy for all the rest of the more complex organisms to have food to develop. And they're the oceanic equivalents of terrestrial plants. So sometimes they form these beautiful blooms. This is a type of plankton that's called the Miliana hax clay, or EHAX. And this is seen from space. And this is a bloom in the English Channel, 1999. However, these blooms are not always good. So sometimes they're detrimental to the environment. This is a bloom in Brittany about 2 years ago. So they have really costly effects to the local communities, and they can be lethal as well. Other components of the ocean is also bacteria, and there are a zillion, I don't know what's the number for this, but there are loads of them in the ocean. And there's also, there are archaea and bacteria. Basically, this is part of another whole debate. The bacterial kingdom is not divided into two bits, the archaea and the bacteria. There are both of them in the oceans. Again, there is a yin-yang thing going on in here. So they can be good, but they can also be evil, especially if they come out of effluents of human interactions. So if they come out of sewage or industrial processes, you can get enterococci or other types of pathogenic bacteria, which have massive effects to us humans. More on this later. But there's very tight regulations in most industrial countries, in the EU, in the US, on how many bacteria there can be in any 1 unit of water. So that's why, because it's so tight, everyone is getting interested how you can measure them, which is good for us. We get money. So. And the most important molecule that is my favorite one is viruses. And there are innumerable of them in the oceans. There's a million in just 1 mil of seawater. So there's many, many, many of them in the whole of the ocean floor. And this is a really cool picture from a Nature paper where you can see this is about 10 microliters of water. So 10 to the minus 6 liters. So it's tiny. It's basically a tiny drop. And they're beautiful. They can be really helpful to ecosystems. This is a really large virus, and it's large because it's about 200 nanometers. So this is 10 to minus 9 meters. So it's small relatively to us, but large as viruses go. And it grows symbiotically with the Miliana haxley, the beautiful bloom I showed you before. And some scientists support that that's the reason why EHAX, this species, has a really evolved genome, because the virus helps it and it keeps evolving and it creates new genes all the time. So they can be really good for the local ecosystems. Again, they can be vicious, so pathogenic viruses, mostly coming from fecal contamination, so again, from us, if we don't have tight constraints of what comes out of sewage plants. And they're pretty resilient, so they can survive up to 2 weeks in water. And they can be polio vectors or rotaviruses, and this is mostly what you can get infectious diarrhea from, so not very pleasant. And also Hep A. So the general type of monitoring, so you can get information on all these different species, is biological environmental monitoring. And the term as such was introduced by a guy called Yves Fouillet from the University of Grenoble about 2 years ago. And basically it encompasses measuring everything. So it doesn't have to be necessarily in the ocean. It can also be in any kind of point of care environment. So it could be at the doctor's surgery. It could be on a beach where someone wants to make a specific measurement, because the challenges, as far as the technology goes, are the same. And the aim is to develop a transportable system which can go from sample collection to delivering the results in one single device. That's a really massive challenge, integration. There are a couple of systems, but they're massive. So one of them is used by the US BioWatch. So in some airports, especially in the States, they have it, but it's massive. It's like 3 tons, and it does everything. So it takes you from the sample to analyzing the results and pop, sort of like CSI, but it's really big. But we want something small and deployable, where it can be used in developed countries like ours, but also in countries where they don't have the equipment to have 3-ton kind of, facilities, like Africa. And you need all kinds of analytical sciences facilities. So you need chemistry, biology, and environmental science, but also system integration and a bit of microfluidics. So I'll introduce you to what microfluidics is. And it's the science and technology of moving liquids into very narrow channels. The channels are usually in the size of the width of a human hair. So this is hundreds of microns. Sorry about this, but it has been squashed. This is an example of a microfluidic device. It's about 5 centimeters in width, and the channels, again, they're in the micron scale, and you can see how they can crisscross, and you can have all this type of inputs, so hundreds of different reagents mixing together in a tiny device. And this was shown by the Whiteside's lab in Harvard that have pioneered microfluidics in general. They have to have also functional elements, so the same thing you would have in large fluidic devices. So they have to have pumps, valves, and columns to manipulate the liquids and force interactions. The first microfluidic devices was shown, it's been a while now, so it's in the late 90s by Northup. But since and that was basically a sort of briefcase with a laptop, and it was a PCR, so polymerase chain reaction. It was doing DNA amplification. Since then, although it's been a while, there haven't been many commercial devices presented. If you've done any molecular biology, you will have seen in the lab the adjuvant bioanalyzer. So this is a microfluidic device, because within it there are microfluidic channels where you can do capillary electrophoresis. So the equivalent of a gel electrophoresis, where you can separate fragments of molecules, usually DNA or proteins, depending to their size. You can do the same thing in these little cartridges. This is one of the few examples, but the whole industry is really growing, and it's being estimated by 2010 that the global market for microfluidic devices will be in the $6 billion mark, which is massive. This is mostly driven by pharmaceuticals for point-of-care diagnostics. So, in order to have information about the different cells in the environment, you need to pick which kind of molecule you're going to target in the different type of cells. So the choice is DNA and RNA. These are nucleic acids, which reside in the nucleus mostly of the cell, and also proteins. DNA gives you information about all cells, so whether they're alive or dead, and genetic information. But RNA is more of a metric of viable cells, because it's really short-lived. So within 20 or 30 minutes after a cell dies, all the RNA will degrade. So basically, this is an information of viability and space, so whether something has been recently present in a specific location. Proteins are more function-specific. So I'm mostly interested in nucleic acids, and this is a diagram of PCR, polymerase chain reaction. So this was invented by a guy called Karamoudis, who got the Nobel Prize in '93 for that. And it was really smart because it uses Taq polymerase to amplify minute amounts of DNA. Usually if you want to target few cells, the DNA there is not enough, so you need to make more of it so you can measure it. And basically, this was made into a patent, and Roche, who bought the patent, made an estimated $2 billion by the time the patent expired in 2005. So there's big money in this. Since then, because this is such an interesting way of studying nucleic acids, there has been a large number of isothermal methods. These methods operate in lower temperatures than PCR, and they're more applicable to microfluidics because they don't have these larger temperatures of 95 degrees and 75 degrees that are necessary for PCR. I won't bore you with all the acronyms, and these people were quite smart, so first they patented all the work, and then they published it. And again, there's been like lots of money made out of all this. And I'm working mostly with two methods. NASBA, nucleic acid sequence-based amplification. This is the one that does the little hump here. So it operates at 65 degrees and then goes down to 41. And also RPA, recombinase polymerase amplification, which works at a constant 37 degrees. So that's really easy to miniaturize as far as temperature goes. So I'll take you through the stuff I've done. This is the work I did at the National Oceanography Center, and it was funded by NERC, but also from a large FP7 consortium. And the aim of that was to study marine algal blooms. This is an example of that, and we targeted Carenia brevis, and this is an example of this cell with the 4 lobes. The reason this is harmful is that it produces these toxins. Toxin A is here shown, and because of this rich polyether rings, it can bioaccumulate and cross the blood-brain barrier with toxic effects to mammals and fish. We targeted rubisco, so one of the most important enzymes in photosynthesis. Our main competitor was NASA, so you know, they won this, I can tell you from the start. So this is a system that was developed at Monterey Bay Aquarium Research Institute from Chris Scholling, and it's quite large, and it measures 1.5 meters. It's integrated, so it can do PCR and also ELISA, which is a way to study proteins. It can be deployed for up to 2 weeks, and it doesn't send the information, it logs it, and then you can pick it up and get everything out after the 2 weeks deployment. So we tried to develop a more portable device. This is not a deployable device, this is more a back of a van type of device. And it has disposable cartridges, and it can be connected via USB to a tablet or an Android phone now. So it has these cartridges, and I can pass you around one so you can see how they look like. Where you can preserve reagents in the chambers. So you have 2 types of chambers. One is for primer annealing, so part of the process that we do at 65 degrees. Another one is for incubation and real-time detection. There are 7 integrated valves and 3 pumps inside the chip. And this is how the internal components look like. There is an integrated confocal detector that takes measurements in up to 2-3 colors, so you can have multiplex detection as well in it. There are 3 peristaltic pumps that actuate the liquids, so push them along inside the chip, and linear actuators that basically are pins that are pushing the valves to move along the liquids. There are also heaters to heat the reaction at the 2 temperatures we need. And these disposable lab carts, where we managed to preserve the reagents for up to 8 months in room temperature. We got quite good results, and we could detect down to 10 cells of this harmful algae. The kinetics of the reaction, this is real-time data, is a bit different to benchtop, but still you can find where the differences are, find correlation factors. The differences in kinetics is because the volumes are so much smaller than you would do in benchtop reactions. So after that, I moved into a position at the Institute of Life Sciences, and this is a collaboration with Public Health England, and this is working with Howell Morgan at Electronics and Computer Science. So we made a simple device to detect infectious diarrhea, which is not pleasant, as you all probably know. And basically, in the developed world, it's really interesting how you get it. Usually, after you've had a hospital stay and you've had a lot of antibiotics, they cause the natural bacteria in your gut to perish. And if you have ingested some spores of microbes, such as C. difficile or Cryptosporidium, they go crazy because they have the gut free to reign, and you start getting toxic effects. So we use the slip chip concept, and I'll show you how this works. So you have two plates made from plain acrylic. Oh, sorry for the quality of this. So you can fill them up by sliding it along. You can create different channels, and you can mix reagents very simply. This has been pioneered by the Smagilov group in Caltech, and basically they just do it with two plain bits of plastic and paper clips, so you don't need anything, really, no power. I made a slightly more complicated device, so I made a holder, but it's still quite cheap and easy. So the most expensive component of this is this caliper that's like 50 pounds. The rest is acrylic that you can rapid prototype using micromachining or laser cutting. And this is how the whole thing looks. There is a fine layer of oil that separates the plates. You can slide them along and move and connect different channels with different reagents. This is the data from the device. So we went down to 1,000 copies of C. difficile, and we targeted one of the toxic metrics, so basically toxin B gene. This is the most potent toxin in C. difficile. And this is how the data looks in benchtop experiments. So this is under submission now. And then this is the more complicated and high-tech devices we've been developing. So this is in collaboration with Sharp Labs Europe. This is the R&D component of Sharp Electronics. And again, Public Health England. This whole thing has been funded by NIHR, and it's with Professor Howell Morgan. And the reason we're doing all this is to target antibiotic resistance. Again, the device you would use for this kind of thing would be the identical one you would use in the environment, just the molecular target changes. The reason we went for antibiotic resistance is because you may know there are three main classes of antibiotics: beta-lactams, cephalosporins, and carbapenems. Carbapenems are the most modern ones. However, antibiotic resistance has developed in all three of them. And basically, this is data from the NHS that we got from Public Health England. And resistance, this is infections of people admitted in the NHS up to 2011. Numbers for 12 and 13 follow the same trend. So resistance to our best and highest technology antibiotics increases almost twofold every year. And there are no more new antibiotics because this type of research is not supported by the pharmaceutical industry. So basically, we are in trouble, is what is the message here. And that's why Dame Sally Davis, the Chief Medical Officer in England, has defined this as a ticking time bomb and that we need to take action. So we're developing devices that could detect the resistance before the doctor gives you the antibiotic, so you would know you don't need to take it and you don't develop further resistance. We're using digital microfluidics. So this is a more high-tech type of microfluidics. And the way this works is you have patterned electrodes on a surface that has been rendered hydrophobic. What this means is that if you put a water droplet on top of it, it will stand proud and tall, and there would be an electrode below it. However, if you apply voltage, the droplet will splay, and if there's another electrode next to it that you then switch on, you're going to make it hop from one electrode to another. And I'm going to show you a video that shows the recombinase polymerase amplification reaction on a digital platform. So you start with reservoirs of droplets. This droplet here is 400 nanoliters, and we have 5 replicates. 3 is the reaction, and 2 are controls. You also have DNA, so what you're actually amplifying in this reaction, and magnesium acetate. This is another reagent that we've pictured here as blue that chemically initiates the reaction. So you need that to get things going. Unless you have that chemical initiation, nothing will happen. So we got our droplets, and then you can mix them by moving them about to different electrodes. This is the chemical initiator. These little droplets are 100 nanoliters, so to -9 liters, they're quite small. The whole reaction volume is 600 nanoliters. So you get this mixing. And you can see this is food dye, so this is not the real reagents. It's clear because it's indium tin oxide, so it's clear patterned electrodes. That's why you can see them. And this is the same technology that you have on your mobile phone screens. That's why you can see through them. And then you have real-time detection above these 5 areas. And you get real-time amplification that you can monitor against a negative control that's specific to that reaction. So you had the same batch of reagents. This is data for E. coli, so we're developing that for E. coli detection. And this shows 80 picograms. We've gone down to 100 copies, so even further down. This is about 500 copies of the genome, of the gene we're targeting. And this is under submission as well. So finally, I'll take you to the last step. I'm planning to move to even simpler devices. So basically, I just got a fellowship, and I'll be moving to Harvard in a couple of months to focus on paper-based microfluidics. And this is using capillary-based microfluidic devices. So the simplest kind of device of that form is the pregnancy test. That thing, but made much more complicated with sample integration. That's what I'd like to do. So these devices were invented by George Whitesides in Harvard in 2007, and they're called 3D micropads. So this is an example of one of them. So you can see different colored liquids crossing each other, but without mixing. And the trick in that is to have complex and really hard to design, really, patterns in 3D layers of paper. These devices are really cheap, so they've been shown and they have been used for a while for detecting hepatitis. And the cost is less than 10 cents per device, including the reagents. So they're really, really low cost. The channels in here are patterned. They can be wax patterned, and there are other ways as well. The plan for me, so this is going back to the carbon cycle, is to use them for water monitoring. So try and detect viruses in the natural environment using paper devices. There are some examples in the literature, so people have used them to detect E. coli in this combination of paper and plastic together. And also, I would like to use them to study viruses as vectors of evolution and how they affect population dynamics in the environment. Because although they're so little, viruses make up to 95% of the abundance of the different organisms in the environment. So we're back to the carbon cycle. So by having the correct type of devices and at the correct locations, particularly if they're really low cost, you could get much more information of how that carbon is circulated on a global scale. So I hope I've convinced you that microfluidics can be used for environmental applications. And isothermal molecular biology methods are particularly good candidates to miniaturize. I've showed you some examples using micro-NASBA and RPA, so two different types of isothermal amplification. And also how we could use paper-based, really low-cost devices in the future. So I'd like to thank my collaborators. I've thanked the funders as we were going through. And finally, I'd like to thank you for your attention. Thank you.